Good evening. Why all of you are sitting that side? Why there's any infection this side? What? Come here, half of you. है कुछ प्रॉब्लम है इस तरफ है क्या? तो आओ ना इधर. Spread up and sit. Yes. What did we study the other day?
We studied about
Cell meaning we must have studied about what? The cell organelles. Did we start? No, I suppose. No? Cell organelles. So we are going to start with the cell organelles today. The parts and the functions we are going to learn. Okay. So the first part of the chapter was about the concept of cell, the history of cell, the discovery of cell. That is the first part of the chapter that you should know. And famous names of some famous biologists, the cell that we are talking about, the fundamental unit of life. That was the chapter. Then we were talking about the contribution of some famous biologists. And if I ask you, what was the contribution of Robert Hooke?
Ma'am, he discovered the cell in the cork.
He coined the term cell. He coined the term cell, and the concept of cells. When we start understanding the concept of cells started long back. Another famous biologist known as Leeuwenhoek, he was the first person to introduce the concept of cells. But at the time, he had studied some cells, he studied some cord tissues, different type of tissues, and the microscope that he used was a very primitive microscope, so nothing was much visible. But he could see that some structures are there, but this was even more put forward very clearly by Robert Hooke by the study of the cork cell. So that was what we must have discussed the other day. So what was the experimental material that Robert Hooke had used? The cork cells, the bark cells. And how did he name the bark cells? How did he name the tissues? What did he name them as?
Little rooms.
Little rooms or cella. He had named them as cella or little rooms, and from the word cella came the concept of cells. Then how did he define what are cells? How did he define? And later the same concept is still what we are studying today. How do we define the word cell? What is the definition of the word cell? Cells are the structural and functional unit of life. Cells are the structural and functional unit of life. So what is the meaning when I say cells are the structural? Did we write down the definition?
Yes.
Cells are the structural functional unit of life. What is the meaning? Cells are the structural unit of life. Meaning, when I talk about the cells, I may talk about the cells of the leaf. The structure of the cells of the leaf will be specific, different from the cells of the stem. The structure of the cells of the stem will be different from the structure of the cells of the root. Why? Because the functions are different. For an animal, if I talk about the structure of cells of the liver would be different from the cells of the intestine. The cells of the intestine will be different from the cells of the kidneys. Why? Because the functions are different. So, what should be the function? What is the function of the cells? Based on that, always the structure. I may understand that the cells of the lungs are a specific type of a function so that they can carry out gaseous exchange. But again, if I talk about the cells of the blood cells, absolute different structure because they have some other function, carry various different type of gases and nutrients and etc. etc. So even in case of the animals, the structure of the cell would always be very much dependent on what type of a function they would perform. So we had already written down the definition. What is the definition of cell? Who coined the term cell? What was the experimental material? So that was the first part. Then after that we studied unicellular, multicellular organisms. Now if I ask you a group of organisms and if I ask you to segregate them, I would say amoeba is there, paramecium is there, and then the bird is there, the duck is there, the fish is there, the bacteria is there. How would you group them? The amoeba and the paramecium and the bacteria all go together. The fish and the bird and the duck will all come together. How? Why are we categorizing them this way? The amoeba and the paramecium and the euclid, they are all unicellular. Whereas the fish and the frog and the duck all go together because they are all multicellular organisms. But now that we talk about unicellular, multicellular, what did we discuss the other day? What did we learn the other day? If it is an amoeba, does the amoeba undergo the process of respiration? Yes. Excretion, nutrition, everything, growth, division. So that one single cell is responsible for all the functions. But if I have to talk about a plant, or an animal, or a lizard, or a frog, or a fish, or a human, in case of a plant, what is the difference? The difference is different group of cells, and then tissues, and then organs, and then systems to perform different functions. Okay, even if I'm not talking about systems and organs, I may talk about a simple invertebrate, I may talk about a simple hydra, I may talk about a simple jellyfish, which may not have well-developed systems, but still they have got different groups of cells to perform nutrition, different groups of cells, tissues to to perform excretion, to perform movement, to perform etc., different functions. This concept of division of the work in the entire organism, they have got different tissues, and the different tissues are meant for performing different functions. This is what we had already written as a point. If it is a multicellular organism, they have got division of work, division of labor. But if it is a unicellular organism, there is nothing known as division of labor. It is one single cell and all the functions. Understood that part? Then next, what was the next part that we studied? Only that much, right? Okay. So next we continue again. Now that was the complexity of the organism that we studied. So far what did we study was the complexity of the organism. Let us now go ahead. I talk about a particular type of a cell. I talk about a cell something like this. I talk about a cell something like this, where this cell has its hereditary material all coiled up, and then the cytoplasm. And in the cytoplasm, the cell would be having just few parts. Some few parts only to perform certain functions. In contrast, I may talk about a well-developed animal cell. I may talk about a well-developed animal cell, and in this animal cell has got similar structures, but these structures are enclosed in a membranous structure. And this membranous structure is also well formed. This membranous structure is also well formed like this. It is also well formed. And all these thread-like structures that I draw here are present here also. All these structures that I draw are present here also, but they are quite distinct now. They are quite distinct. What about the parts outside the central region? Okay, the parts outside the central region also too many. They are of so many different types. And now that I talk about, then which type of a cell is known to be complex, which is a simple type of a cell? How will we understand? In this cell, if you see, it has got fine thread-like structures, and these fine thread-like structures are known as the chromatin fibers. Does this cell also have chromatin fibers? Yes, these are the chromatin fibers. Now let us talk on this particular concept. This cell also has chromatin fibers. This cell also has chromatin fibers. I would say this is a bacteria. I'm saying that this is an animal cell. So we understand very well that this is a bacteria, meaning this must be a simple cell, and this is an animal cell, meaning this must be a complex cell. But how are we convinced that this is a complex cell? It is a complex cell because these hereditary units, the fine thread-like structures in the central region of the cell, are covered or wrapped up by a well-developed membrane. And there is a distinct region known as the nucleus and a distinct region known as the cytoplasm. And these thread-like structures known as the chromatin fibers, they are only centrally placed, or if I have to talk about a plant cell, they would be a little bit away from the center, but they are not spread out in the cytoplasm all throughout. That means whether I talk about an animal cell or a plant cell, the nucleus may be either in the center or a bit away from the center. But one feature that is common is that yes, they may have the nucleus bit away from the center. They may have the nucleus bit away from the center, but whatever it is, they will distinctly have something known as the nucleus and the cytoplasm, and with many such inclusions here. Try understanding the major feature. The major feature is chromatin scattered in the cytoplasm, chromatin not scattered in the cytoplasm. Chromatin covered by double-walled membranes, not covered by any membrane. So, if the chromatin is not covered by any membrane, then can we say that this is the nuclear region? And no, where is the nucleus and where is the cytoplasm? The chromatin is all spread out. There is nothing known as nucleus and cytoplasm for this cell, but it has got chromatin material, either one or two. Then what are we going to name this chromatin material that comprises a major region of the cell? We would say such type of a nuclear region is actually known as the nucleoid. Nucleus, nucleoid. So yes, this is a double wall structure that I'm talking about and well formed. This is a double wall structure that I'm talking about and well formed. Same here, same there. So then what type of a cell it is? Is it common? Are they of the same category? Yes, they are of the same category because they have the nuclear material surrounded by well formed membranes. Such type of cells are known as eukaryotic cells. These are known as eukaryotic cells. The word "eu" refers to the advanced cells. They are advanced. How advanced? Because the nuclear region is all concentrated, covered by one protective membrane. The cell membrane, nuclear membrane, will come to this later. So there's a protective membrane differentiating the cell into two parts, the nucleus and the cytoplasm. Nucleus and the cytoplasm. What about this cell? This is a prokaryotic cell, meaning this is a very primitive cell. Why primitive? Because it does not have any well-developed nucleus. There is nothing known as nucleus, and since it has got a concentration of the hereditary material, we will say this is a nucleoid. What about the cell inclusions? Do they have cell inclusions? Meaning, do they have enough number of cell parts? No, they don't have enough number of cell parts. They may have few parts known as, just few, known as mitochondria. Or they may have some granular structures known as ribosomes. That's it, nothing apart from this. And what about this? They may have the ribosomes and the mitochondria and the Golgi and the endoplasmic reticulum and the lysosomes and so many more. So definitely this is a complex cell. This is a complex cell. Then what is the major difference you are supposed to understand now? What is the major difference you are going to write now in your exam? Difference between prokaryotic and eukaryotic cell. What is it? If it is a prokaryotic cell, it does not have a well-formed nucleus. Then what are these unorganized nucleus known as? This unorganized nucleus known as nucleoid. Prokaryotes, how are they different here? How are these eukaryotes different here? The eukaryotes, they will always have a well-formed nucleus surrounded by the nuclear membrane. Okay, let us talk about one more difference. I said chromatin fibers. Yes, both the type of cells have. But if the chromatin fibers of this type of a cell, something like the bacteria, how many chromatin fibers? At the most one or two. What about this side, plants? What would be the number? The number is variable. It may be 2, it may be 8, it may be 16, it may be 32, it may be 38, it may be 46. So many. Depending on which organism we are talking about, the number of chromosomes is not a constant number at all. The number of chromosomes of the onion and the grasshopper and the honeybee and the palm trees and the coconut and the fish and the frog, all different. The number of chromosomes for every type of organism, plant or animal, different. But the moment I go to this type of cells, and this is a small fraction of organisms that we understand, prokaryotic and eukaryotic. So what did you learn? What is the summary to understand? Summary is, if it is a prokaryotic cell, it is a very simple cell. Eukaryotic cell, it's a complex cell. Prokaryotic, nucleus, well developed or not developed? Not developed. What is that underdeveloped nucleus known as? Nucleoid. And in case of a eukaryote, what is the characteristic of the nucleus? Hereditary material surrounded by the nuclear envelope, the nucleolus, everything, all parts are there. What about the chromatin? How to differentiate? Chromatin material present at the most 1 or at the most 2, not more than that ever. Whichever species of bacteria you talk about, bacteria also present in so many types, no? But whatever types of bacteria you talk about, the number of chromatin will never be 5 or 10 or 15 or 20, never, 1 at the most 2. What about this side? Plants and animals, the number of chromosomes, many and variable. Okay, many definitely, but variable. We'll first write one difference between these two types of cells, prokaryotic and eukaryotic. Write one question. Make a table neat. Differentiate between prokaryotic and eukaryotic cells. Major three differences you should know. Differentiate between prokaryotic and eukaryotic cells. Differentiate between prokaryotic and eukaryotic cells. Make a table. Differentiate between prokaryotic and eukaryotic cells. Under prokaryotic, the first point, under prokaryotic, right? They are primitive cells. Primitive cells with an unorganized nucleus. Primitive cells with unorganized nucleus. Unorganized nucleus. Primitive cells with unorganized nucleus known as nucleoid. Other side, eukaryotic. These cells have a well-formed nucleus. These cells have a well-formed nucleus. Where the hereditary material, that is chromatin fibers. Chromatin fibers are present inside present in the cut that word inside. Present in the central region of the cell, in most of the cells. Present in the central region of the cell, surrounded by nuclear membrane. Surrounded by nuclear membrane. Surrounded by nuclear membrane. Next point, number 2. Prokaryotic, they contain nail. Second point you should always write from here. Where the first point ends, then next line should be your second point. Second difference, prokaryotic, chromatin fibers. Chromatin fibers are 1 or 2 in number. Chromatin fibers are 1 or 2 in number and are extensively long thin and coiled. Extensively long, thin and coiled. Other side, eukaryotic. Chromatin fibers are coiled to form distinct chromosomes. Chromatin fibers are coiled to form distinct chromosomes. Are coiled to form distinct chromosomes, comma, many in number. Are you understanding? I'll give you time to read. So see that you are able to understand. Wherever there's a doubt, you have to clarify. Chromatin fibers are coiled to form chromosomes, many in number. One more point we'll have to write. Number 3. The third difference. No, this is not the way. Which school? Huh? No, why do you write differences this way? Difference you are not supposed to write this way. This is 1 point, 1 point. Where do I start my second point? I will start my second point should always be parallel. This is my second point, this is my second point. Cut this and write here. Second, see there. See here, switch. Exactly. Second point should go there. Same school? So?
Same class.
Then? How come you are writing this way?
I almost read it this way.
Nay, this is not the way. This is not the way. Your teacher must be loving you very much. That's why she keeps it on. That is not the way. Write proper pattern format should be exactly the same. Otherwise, the same thing you will do in the 10th. ये habit बन जाएगा. Okay, now chromosomes. The third point we will write is third point under prokaryotes, right? They contain few mitochondria. They contain few mitochondria and ribosomes. They contain few mitochondria and ribosomes. To carry out cell functions. To carry out cell functions. To carry out cell functions. Other side, eukaryotic cell. Number 3, variety of cell organelles. Variety of cell organelles like mitochondria, ribosomes, Golgi complexes, lysosomes, etc. Variety of cell organelles, mitochondria, Golgi complex, ribosomes, lysosomes, etc. carry out cell functions. Carry out cell functions. Carry out cell functions. So now, this is basic to understand prokaryotic primitive cells. What are the characteristics? Advanced cells. What are the characteristics? Now next, we'll have to try understanding what are the characteristics of a plant cell, animal cell. Plant cell I will take later. Once you are thorough with an animal cell, understanding plant cell is very easy because there are quite a number of parts that the plant cell shares with the animal cell except for 2-3 parts. So we will start with the animal cell. All of you have pencils with you? Yes or no? Pencils? Now take out your pencils. And in your notebook, now the next page, in your notebook, in half of the page, you will draw this diagram. Now, we will not draw this diagram at one go. Part by part, as I draw, as I explain, you should be acquainted. This is this part, these are the sums. This is this part. So that's how I will make you draw. First, you draw this in half of your page there. Not too big, not huge, large, but not too small. Now, if this is my ruled notebook, I will always diagram drawing. Now, in the 9th, 10th, we have got some marks for diagram drawing also. Always try to pick up full marks for the diagram drawing. 2 marks, 3 marks for it. This is the page. This is the left margin that you have. Your diagram should be little bit towards the left, so that I have enough space on the right, because I have to put all my labels as much possible on the right. Pencil only to be used, no pen, no color, nothing. And diagram should be little bit neat, easy to get full marks there. How big should be the diagram? This is a ruled notebook, so diagram should be minimum 12 to 14 lines. So that should be, because it has got many internal parts to be drawn. You draw a diagram small, you'll be making the diagram too congested. So minimum 12-15 lines should be your diagram. Draw the diagram little bit towards left, as I mentioned just now, so that there is enough space in the right to label all parts. Draw this diagram and label the plasma membrane as of now. The nucleus I was just explaining, so I had labeled. Only draw this much and label the plasma membrane so far. Don't make the diagram absolutely this one rectangular. Rectangular cells are majorly plant cells. So it can be little bit oblong, spherical, not round exactly. Scale. Freehand. This is not the shape. When you draw this shape, this as a shape in your exam, marks may be deducted. What should be the shape of the cell? This will be the shape of the cell. Little bit oblong, not exactly square, not exactly rectangle. It should not be a